So hi, hello everyone, and welcome to another Saturday microscopy live stream. Microbehunter here, Oliver here. And yes, look, these are all little larvae of Artemia brine shrimp, also known as sea monkeys. And today I simply wanted to play around with them a little bit. I want to show you how I have grown them out of eggs. Yeah, and we can put them under the microscope, and yeah, maybe I'm also able to make a couple of prepared slides. So for those of you who are new here and those of you who are not new here, it's become a tradition in this live stream that you, if you want to, of course, that you quickly introduce yourself and from where you are, because microscopy is a hobby that is enjoyed all over the world, and it would be also nice if you could, someone maybe could quickly inform me also in the chat if the sound works and if you're able to hear me well, or if there's some kind of a distortion here. Yeah, and I see that there are already a couple of comments here, all the way from New Zealand, from Virginia in the United States, from Scotland, from the Netherlands, from the UK, from the Netherlands, from India, from Honolulu, Hawaii. Hi Oliver, I'm over in Denmark. Yep, Australia, sound works fine. That's always nice. Okay. Evening from the UK, sound is okay. Sound is good. Florida, from Berlin in Germany. Yeah, welcome everyone. What I usually do is the first couple of minutes, I'm really usually not diving in directly into the topic because there are in the first 5 minutes or so more and more people joining in. I checked the statistics, and at the beginning usually there are only very few people, and then yeah, more and more people start to join in. So that's why I want to yeah, wait a little bit with the start here. Okay, yeah, greetings from Berlin and Romania. Okay, yeah, it's all very nice. Yeah, would you please consider doing some videos analyzing and discussing prepared slides with respect to histology, anatomy, etc., as if you were teaching biology to higher college class? Yes, there are some specific requests as well, and the answer is yes. So the question is if I would not only do show you some slide preparation, but actually show some histology slides. Histology, these are our tissue cross sections, and talk about those. And as a matter of fact, if you're interested in this type of direction of microscopy, then I would like to invite you to visit my other microscopy channel. It's called simply Microbehunter. It's actually larger than the one that you're looking at right now, and I've actually put some histological slides there under the microscope as well, where I basically do a little bit something like a biology lesson. Yeah, and so I'm actually doing that, but not so much in this channel here. Yeah, hello from Spain. Yeah, and those monkeys, the sea monkeys, are adorable. Yes, they are. And what I'm going to do now is I'm going to simply start, yeah, talking a little bit here right now, and yeah. Then we can also try to make some prepared slides. Now, a short background information about those little sea monkeys. Those sea monkeys, that's basically how they are sold to children. They can be bought in a kit, and normally, not normally, but the real reason why those little brine shrimp are actually sold is because people who have an aquarium, especially saltwater aquariums, they grow them as fish food. So they are actually bred in large quantities for fish food. But at the same time, they're also selling those for children, so the children have something to look at. But the larvae that you see over here, they're pretty small, and for this reason, I think they're a very nice specimen for microscopy as well. And I'm going to just show you how you prepare them. So if you're interested yourself in actually breeding them at home and playing around with them a little bit, or if you have children who are interested in doing that, I'm going to just show it to you, because I think that actually can be combined quite nicely with microscopy here. Okay, yeah, so hi from Lithuania and from Texas, USA. Okay, also, so what I'm gonna do now is yeah, I'm going to switch over to the desk view again, and I'm going to just show you a little bit of what I've got here. Gotta reach all the way back. What I did is I ordered myself. Okay, so that's basically yeah, it's from they sell them all over the world, but this one is just happens to be the German kit that I got, and it comes in a box like this, and yeah, depending on what you buy, a whole bunch of more or less useful things come along with it. yeah, to keep children happy, but actually that's not really so important. What is important are the instructions, obviously, and in this box there was yeah, something like this here, okay, and yeah, it basically there's a fish on here. So why in the world is there a fish on here if I actually want to grow those little tiny little brine shrimp? Yeah, because this is actually sold as fish food. And what you've got in this pack here, so if you just open it up here and you look into it, I'm just going to show this to you. Yeah, you're going to see that there is some powder in here, and this powder actually has two colors. You see there's a white color and a brown, color here as well. And the white one, that is salt. And the brownish colored powder, these are eggs. So there are, I don't know, a large quantity of eggs and salt here. And what you have to do is you have to mix this mixture with the correct amount of water. The water should not be chlorinated, of course. So if you have chlorinated tap water, then you have to boil it and allow the chlorine to escape. And then, of course, you have to let it cool down to room temperature. And then what you do is you take a little... I bumped into my camera here. You take something like this is what happened to be included. And you take a mixture here and you put it into the correct volume of water. Now, when I tried this the first time, it did not work because I made a mistake. You see, if you shake it a little bit, you see that the salt and the eggs, they kind of separated a little bit because they have a different density. And what I've done wrong is I only took the eggs and I took too little salt, and then they did not hatch. So what you should do is you should kind of mix it properly, and get yourself a small, scoopful of eggs and salt, and it's probably more salt here than eggs. Okay, you get yourself a little scoop here, and what you have to do is you have to add it to the correct amount of water. I'm going to show. I just prepared some tap water here. It says 20 milliliters of tap water for this amount of mixture, and so that's regular tap water, and that's it, okay? You probably want to shake it a little bit. And then it says that you have to wait approximately 24 to 48 hours, and then you're going to hatch. It also says, the instructions also said that they like light, so you should not keep it in complete darkness, and it should not be too cold. So they actually recommend about 25 degrees centigrade. I don't know how many Fahrenheit that is. It's a little bit warmer than room temperature. And and it should be bright. It shouldn't be completely dark. And then, and this is basically what I've done here, and then after about 24 to 48 hours, and this is something that you might have problems seeing, there are a few hundred tiny larvae in here. What they will do is they will eat. They come along with. They're able to survive several days this way, and then you have to add a little bit of food if you want to continue to grow them. If you want to have more water, more liquid, like for example, you would like to add it into a larger jar, then you have to mix in the correct proportion of non-iodinized salt. In many cases, the salt contains iodine. They added some iodine, but you have to take some sea salt without iodine, and you have to mix in the proper amount, and then you can essentially simply dump it over into a larger jar. Okay, so this is kind of the thing, and what I'm going to do, what I've done is I've simply waited now 2 or 3 days, and I took a small amount of those of the water sample and put it on a microscope slide, and this is I'm going to just show you how I've done this, and this is where I got something like this here. Okay, so those large round structures, these are of course the eggs. What I've not seen unfortunately yet is how they actually hatch out of the eggs. I was not lucky enough, but maybe, I don't know, if you look over here, so you see that some of the eggs are not completely round. Okay, yeah, so maybe there is some hatching going on. Now, you might wonder a little bit about the rainbow colors that you see here. And this is because of the optics that I have. So if you actually look at those under the microscope, you're probably going to see it more like this. Okay, so that is the normal way that you would see it. Yeah, but I added simply a couple of colors. So that is a regular bright field right now, and I'm using the lowest power objective, so I'm just using the 4x objective, and then you're able to see that, see it like that. I did not add a cover glass because I did not want to impair the movement of those little larvae, but I'm going to add a cover glass later on because I would like to increase the magnification. And but But right now you can see over here, the microscope slide here in the corner, there is a large enough distance, so there's no danger for me touching the water, and for this reason, it's safe. Otherwise, you should actually add a cover glass under normal circumstances. Okay. What do you have to feed them? This is now the next question. Yes, that's a very interesting, important question. Indeed, there is what I have fed them. There is something I'm going to show you. I've got two possibilities here. Now you see it. This is dried yeast, baker's yeast, the dried form. And all I've done is I added a little bit of, I'll just show you. Okay, here, I'm just going to show you how this looks like. I mean, you probably know how dried yeast looks like. That's simply some powder. Yeah. Okay, so that's how it looks like. Yeah. It dissolves in water, and they have in the instructions they have warned that you should not overfeed them. That's really important. Yeah. So I'm actually using dry yeast to feed it to them, and also to other water fleas, for example. However, then that's where it's getting interesting now. People who actually grow them in very large amounts for fish food, there is actually, look what I got here. I'll just show you also the original package. Again, this is, they actually have basically food, liquid food, for those, for those shrimp. Okay, liquid starter food, it's called. Yeah. And I got this again from an aquarium shop. This is already very old, really. But the instruction manual actually said that, it stores, it basically stores almost indefinitely. Now, I opened it just a couple of minutes ago because it's the first time that I'm actually using this. There is actually some food here, and it's green because there's some algae in there. And in a very small amount, so that they're able to eat it. And I'm going to just feed them so that you can actually see it as well. Yeah. So that's basically what has been recommended. And what they also said is you have to add very, very little of this. So just that the water becomes a little bit cloudy, and then you have to wait a few days for the cloudiness to disappear, and then you can add more food. Okay, but that's basically how it looks like. Yeah? So I can either add some yeast or some of this here. Yeah, I've found out that it's very concentrated. Okay, yeah. So there have been a few more questions here for people who just joined in. Okay, so first of all, there was a hello from Butler. In the United States. Hi, hello. Yeah, I just talked about what you have to feed them and what kind of animals are these, insects or crustaceans. They are crustaceans. So they're related to the crabs. They're shrimp, basically. They're brine shrimp. So these are saltwater shrimp. And for those of you who joined in just right now, they're branded and sold and marketed as so-called sea monkeys for children. And I just also want to show you something else. If you really want to have a large quantity of them, look what I got here. Yeah. Oh, I got to change over to the desk view again. Here it is. Yeah. Look, they sell them in huge quantities as well. And this here is just sea salt with food for those. Yeah. I have not even opened them yet. I got these, I think, over 10 years ago. I've never used them because I simply didn't need that much quantity, because I wanted to do some experiments with my students in school, but it turned out that this is way too much, and as a matter of fact, I kept them closed, and something like this is enough. Yeah, so that's basically what I've done. And I'm going to just basically show you now how I take them out. And I also have over here, here show it to you. You might not, maybe you've seen it already last week if you joined in. These are adult ones. And they're pretty huge already, and I bought them in an adult form, also from an aquarium shop. So they're also selling those basically ready-made, so that you don't have to breed them yourself. Yeah. Okay. How is it possible that they can survive like that in a jar for such a long time? Well, they are able to survive because I frequently open it to allow fresh air to come in. That's very important. And as a general rule, and it doesn't really matter what you keep in a jar, I put the lid on top because in case, so that don't spill it over. But I just want to show you something over here. Also, here are some plant material from a pond water. Every time when you put a lid on top, and that's really important, make sure that there is enough air inside the jar. That's really important. And frequently open it to allow the air to exchange. I did some time ago, I made a mistake. I was traveling and I took some of those containers along and I picked up water samples and I filled them up all the way to the top and I put a lid on top, and then there is simply too little oxygen in there and it starts to decompose. It starts to turn bad. It starts to turn anaerobic, as we say, and then everything's gone. Okay, everything's gonna die off. So if you want to put a lid on top, make sure that there is plenty of air inside the container. It's a really important. Ideally, you can leave it open, and when the water evaporates, you simply add some more water. That's probably even better. Okay, and I don't know. This sample here is weeks old already. Okay, so if you make sure that it doesn't completely evaporate, then you can store it for a very long time. And those animals here, how are they able to survive? Well, you occasionally have to add a little bit of food. And of course, they're not going to survive forever. So that's also something completely normal. And for this reason, I suggest if you want to do this either as a hobby at home or with children, then I suggest that you always make small amounts, and because a pack like this is going to last forever. Okay, so I'm gonna go again through a couple of the questions here. Okay, these are very small shrimp. Yes, they're quite small. Yeah, so that was also an answer to one of the questions. I had a kid when I was a kid. The decorative top did not latch, and my cat knocked them over. And that is one of the reasons why I always put a lid on top, so that you don't, when you knock them over, that not everything is lost. But then again, you got to be a little careful that you, it doesn't turn bad. So what I'm going to do is I'm just going to simply repeat that what I've done before. I just wanted to tell you the following here, and that's also important. It doesn't matter which pipette you use. I mean, these are pretty cool looking ones. I already made videos on this. Of course, also plastic pipettes like this work. Those disposable pipettes, you have to look a little bit, is how big is the hole here. Okay, so for the tiny larvae that I have in here, this hole is large enough. Okay, and if you're using a pipette here, and what I've done here is I used scissors to cut the tip off to make it a little bit larger, because every time when you suck in something through a very small hole, there are very strong shear forces and it might actually damage them. So I simply cut it off the tip, and because this is a disposable tip, you just press this button here, and then this one is it's kind of thrown off and ejected. But it doesn't really matter what you use. And what I found out, however, is that there are quite a few of them on the bottom, because all of the eggs kind of settle to the bottom. And I found many of them also gathering on the bottom, maybe because this is also where the food is as it settles. However, some of them might also kind of swim to the top where the oxygen is. So we're just gonna try our best. And what I simply do is I go in here and I'm going to try to suck up a couple of those eggs and whatever I was able to catch. Okay, and you know what? I'm just gonna put it under the microscope as it is, without a cover glass, simply to check, and then I can always add a cover glass. Okay, I'm going to close it again. Don't want to knock it over, and I'm going to switch over to the microscope view again. And we should actually be able to see the same thing here, because so over here, what do we of course, as expected, the very same thing. Some of here they are again. Okay, more or less moving. And what I'm going to do now is the following. I'm just going to add a cover glass. Now, every time when you add a cover glass, of course, you have to there is the risk of kind of squashing them. And for this reason, we just have to be aware of that. So maybe I'm going to add a little bit more water. I don't know. It depends also on the size of the cover glass. If the cover glass is very large, I've got huge cover glasses over here. I mean, very convenient. Look, they're pretty large. They're almost covering the whole slide. Then, of course, the amount of water is going to form a very... the water might not be very much, and the film might be very thin, which might squash them a little bit and limit their movement, which might be an advantage, actually, sometimes. You know what, I'm just going to maybe add a little bit more liquid, and then I'm going to try the large cover glass. Okay, and if there is too much liquid, then I can always remove a little bit using some tissue paper. Okay, so let's have a look here. let's spread it apart a little bit. Yeah. And let's give this here a try. I have to tell you the following, a small disadvantage of those large cover glasses. You see those cover glasses, I don't know if you're able to see this quite well, but they go all the way to the edge. So this means that the water spreads all the way to the edge, and what I've actually found out also is sometimes that the water then is able to spill over a little bit, and then the stage of the microscope becomes all wet. Wow, there are lots of questions right now, so maybe I'm going to just read those and respond to them. The eggs have naturally evolved in a way that allows them to nearly dry out completely and to wait for the most optimum conditions. Yes. In this dry state, they can store for years. What I heard or read, however, is, I don't know about this, that they should not become too hot. So you should not leave, I don't know why this would be, because in the natural environment there's also sun, and they should also be able to survive in a very dry lake. But I heard that the temperature might harm them. Something that one might experiment with. Can one raise them to reproduce? I see no reason why not. The company that made those actually raised them to reproduce. So if you're able to get the conditions right, I see no reason why it should not work. I've not tried it, however. Okay. Is the diet just yeast sufficient? I don't know about that. Yeast is probably not the normal diet, the normal food that they would eat. Okay, and because in the natural environment they eat algae, that's why they have this liquid algae solution here, algae food here. But I simply read that the yeast should also work. Yeah? Okay, I guess they're not very specific here. Yeah, and including chlorella. Or spirulina or other algae. Yeah, I think what I read is that those algae like spirulina and chlorella are actually included also in here. That's why it's green. We're going to have a look at this later. Okay. Could you use phase contrast? Problem. Yeah, I can do that. I'm going to show you, but the problem is I only have a 40 times phase contrast objective and the magnification is very high. Okay, so this is a little bit the disadvantage. So this is now the one with the cover glass, and because everything is spread apart, I wonder where they are. Up here is one. Okay, here is one, and this now allows me to actually go up with the magnification. Okay, and then I can also show you some phase contrast. Okay. So if you were kind of wondering about the blue background, this is because of the optics that I have. It's called DIC. Yeah. Yeah. And if you just want to see it in bright field, I just have to remove the filter and go down a little bit with the light. And that's basically regular bright field like you should be able to see without problems. You see that the cover glass does have the advantage that it limits the movement a little bit. So it's easier to observe. So otherwise, this little critter would kind of whiz all over the place. So 20 times, I'm going to back. DIC takes a lot of light. Okay. So I also have to adjust the other filter. Yeah. And I'm going to show you the adult ones later on as well. The adult ones, they basically look, they're huge. Okay. Yeah. And yeah, I'm going to go here. Over here again to the questions. Yeah, so phase contrast I'm going to show you in a minute or so. I've bred Artemia salina. That's basically the name of this these sea monkeys here for a few years, and I noticed the mature ones often play with sand particles. Never seen that. Interesting. Maybe that's something they'll do with the eggs as well. This could be, or maybe could they be digging for food? Maybe, I don't know. It's interesting. Yeah. How large are these monkeys? They must be pretty small if they fit in the tiny water. Yes, the ones that over here are tiny, less than a millimeter. However, the adult ones, which I'm going to show you later, they're pretty huge. They're this size. Okay, yeah, and they look different. So these are the larvae stages here. Yeah, they must be pretty. Yeah, so they are pretty small, and as you can see, the cover glass does not squash them completely. Yeah. It limits their movement. So I'm going to go now to 40 times. And every time, as you see, I have to go up with the light intensity a little bit. And at that magnification, yeah, it already becomes difficult to see because, yeah, of the movement here. So there has been the request phase contrast. Okay, you're probably going to be disappointed. Okay, I just want to make this clear because So I have to go all the way down. This is my phase contrast objective, and I have to put in the phase contrast filter. That's the wrong one here, and I have to open it, and that's phase contrast. Okay, let me remove the other one over here. Let's go down a little bit. So that's phase contrast. Not you see, water organisms are too thick for phase contrast. Phase contrast is really good for very, very thin specimens, like for example bacteria. Yeah? But yeah, so you see, not very good. Kind of shows that different contrasting techniques are better, best for different things here. Okay, I only have 40 times phase contrast on here, so I think we're just going to switch back again. Okay. Yeah, that's the dark field. If you use the phase contrast filter, the phase contrast annulus. That's yeah, in the condenser, with the 4 times you get basically you get a dark field, but this only works from a 4 times objective. Yeah. And let's go back, I would say, to the regular. Okay, let's go back to the regular one. I have to close the condenser here. Here we are again. Okay. I would not use yeast long term. There's dry food algae. You can feed them. After a while, they start to grow colonies, and the algae feed from the waste. Yeah. Okay. Yeah. Again, if yeast is, I would say, is not the natural food, because why should there be yeast in the marine water where they're swimming around? Yeah. So the little guy is rowing. Yeah, he's not getting anywhere. That is correct, because I think the reason could be why they're not getting anywhere is because the body itself is thicker, and it could be that there is pressure from the top and bottom. So it's kind of he's not able to move forward. Yeah. And so I think this is the reason. Okay. Can you see the egg sac in the newly hatched? I don't know. I don't know if we're able to see some empty eggs here. Yeah, I guess it's a question of luck of actually finding somebody, or I just go down with the magnification. I have to go, yeah, and then maybe we're able. I don't know if this one in the center here. Some of them are a little bit more oval than others. Look! Maybe look. Could it be that there's something? I don't know about those here. Could it be that these? Look, this one might be hatching. I saw some movement here. That's nice. I've not seen this before yet. Okay. Look. Maybe this guy just came out from the egg. I don't know. And what about this one over here? Not moving. I wonder which egg it came from. Maybe the one on the left. I don't know. Would be kind of interesting to see. Okay. So a couple of more questions here. You should also add stuff like clean stones, etc., where algae can grow. Yeah, I think that's really important generally. If you want to, I admit, about 20 years ago, a long time ago, I had an aquarium myself, a proper one, and it's really important to have not only plants in there but also rocks and stones, because it is the place where microorganisms are growing, and they're feeding on microorganisms. Also algae, of course, grow on the biofilm on the surface of rocks and stones, and also of course on the window of the aquarium. Yeah, so it's important to have a sufficiently large surface area. Would they be more nutritious as food source with the egg sac, especially since the food we give may not be as balanced? I don't know. For the fish. Yeah? What I assume, however, I mean, I'm hypothesizing now, is that if you kind of throw the eggs into the water, of course some fish are going to eat them right away, but I guess they're going to settle down into the ground, and maybe they're going to not be accessible to the fish anymore. And then those, the larvae, however, they're able to swim around, and they're able to maybe grow, of course, as they eat up other microbes. And then the fish eat them. So it's like plankton. Yeah. So that's actually, I've not seen this before here myself. Let's switch on the thing here. Yeah, it's again with the DIC. You can play around a little bit with the filters here to give it a little bit more color. Yeah. So that's basically how it looks like. What I'm going to do now, what I wanted to do as well, and that's something I've not tried yet, is I want to actually try to also make a permanent slide of them. Yep. This actually means that a few of them are not going to survive the procedure. Okay, because I would like to test a little bit to what extent yeah, the mounting medium that I'm going to use, which is glue, is actually able to maintain the shape of them. Okay, so I'm just going to show this to you, what I would like to do. I'm going to switch back now to the desk view, and then I'm going to later on, I'm also going to feed them. Yeah, I already made videos of this. I got this from a recommendation from one of my viewers. And then I did a little bit of research and I found out, yeah, it actually is suitable. There's this glue here. It's a water-based clear glue called Elmer's glue. They also have a white glue, but this one is a clear one. And this contains a substance called PVA, polyvinyl acetate or polyvinyl alcohol, or a mixture of this. And indeed, PVA is a documented mounting medium. And apparently it is also in this glue over here. And I said, wow, cool. So I'm going to try it out. And as a matter of fact, it does work. I'm not saying that it's an ideal one for all specimens, but it has one significant advantage which other mounting media do not have, and that is that it is water-based. Because if I were to add now a mounting medium that contains a solvent, then in many cases the solvent in the mounting medium will, for example, alcohol or something like that, will actually withdraw the water from the water microorganisms, the Artemia, the salt brine shrimp, and it causes them to shrink. So that is a problem, but so I'm kind of a little bit hopeful that maybe a water-based glue actually will work. Okay, so I'm going to try to do that. Okay. Yeah, again, a couple of comments. I always try to interrupt myself here. The eggs have a hard shell that some fish cannot pass through the digestive tract. Oh, that's interesting. Always hatch and separate when feeding to small fish. Larger fish are okay, but will prefer flake. Okay, good to know. So the eggs apparently, yeah, are resistant to being digested. Yeah. Where am I from? I am doing the live stream now. Didn't introduce myself, and that's from Europe, from Austria in Europe. So this is basically where I am from. So right now it's Saturday evening, late in the evening on Saturday, where I am from. Yeah. Can you make a video debunking fake microscope videos and channels that spread misinformation? There are a few of those. I was thinking about that. Yeah, fake. It depends a little bit what the fake microscopes are. There has been historically a fake microscope where back in the, I don't know, in the day where the person claimed that he's able. It's called what? Yeah, he was able to see viruses with microscopes. And I think was it the Reed microscope or something like that. There was even an article in Popular Science about that. Yeah, but which is not possible to go beyond the resolution limit. Yeah? So it's actually something I have to research a little bit. Okay. Beste Grüße aus Wien, beste Grüße aus Oberösterreich. I had to say something in German now because there have been greetings now in in German from someone who is also from the same place where near where I'm from. Okay, so what I'm going to do is the following. I'm going to now do the follow. I've got here. If you look very carefully, I think I've got 3 or 4 of those little, how do you call them? Yeah, larvae. Now lost my words here. I'm just going to show them to you because I would like to now do the following. I have to find them. Here they are. Okay, here they are. Okay. And here they are. And what I have to do now is I have to add some glue on top and a cover glass. I know it's not nice, but it's for the sake of science. Okay, so I have to preserve them now in some glue. So what I'm going to do now is I'm going to simply add some glue here. The remaining water should basically be removed by the water-based glue, and then I'm going to add a cover glass. Okay. So let's have a look here. So here we are again. Okay. I meant the newly hacked shrimp with egg sacs as opposed to later on when they have been used. Ah, okay. Is anyone viewing this as a child? If this has existed when I was 7 to 12 years old, I would have been over the moon. I hope people share this with their children who might be scientifically interested. Yes, by the way, the video, of course, is going to be afterwards, of course, also online in this channel. So you simply can watch them later on. And those of you who are not able to join the live stream, of course, can also watch it later on. So what I've done is the following. I simply filled in some of the glue over here into a separate container. And let's try this. Ooh, see? Ah, that's the problem. The glue glued the cap. Okay? So, okay, I cannot open it because it's completely closed. There was some glue left over and the glue was so strong that I cannot remove the cap anymore. But you know what? It doesn't matter because I'm just going to use the glue directly from the bottle here. Okay, all you do is you open this here a little bit. And yeah, I've been thinking maybe I should wait until it's completely dry. But you see, if it dries up completely, then there is the problem that, again, it starts to shrink and shrivel. So yeah, we're gonna see. And by the way, next week I would like to put it again under the microscope to see how well the slide was actually conserved, or if there's any change to the shape of those little larvae. Okay, yeah, and for those of you who joined late, I will show you also adult ones. Okay, so what I'm gonna do now is I'm just gonna put on the cover glass. Okay, and let capillary action do the rest. So you see that the glue slowly spreads. And now you should not put it under the microscope because something might happen, which happened to me several years ago. And that is I was so impatient and some of the glue, or the mounting medium in that case, spilled out on the side, and so much that I accidentally rotated the objective into the mounting medium, and then I had to carefully clean everything off again. Yeah, I think it's fine that I will try it because I will not be using too high of a magnification. But let's see, and I have not tried this before, so I do not know what to expect. But let's try to see how this looks like now. So I carefully remove this here again, and let's put this in here. And now I have to find them again, and hoping, I don't know if that's nothing. I have no idea what this is. So that's those little circles that you see. These are bubbles. Okay. I will remove have to always flip out the condenser for the low power. And now let's see if I'm able to find them, because sometimes what happens is if you put them into the center and if you then add the cover glass, it spreads out all over the place. And you know what? I'm just gonna go over and look directly through the microscope very quickly if I'm able to find them. I don't know where they are. There were 3 of them. But not very successful finding them. You know what I have to do? Oh, I found one. See, and this can happen. Here it is. This can happen because when you put them in the center, you put a drop of glue on it, you put the cover glass on, and everything is pushed to the side. Yeah. And here this one is pushed right to the edge of the cover glass. Huh. Okay. So this actually shows that the strategy was not a very good one. What do we have here? Yeah, see, the other one was also pushed to the side. And the third one here, and the fourth one. See, that's actually, this actually is a nice demonstration on what not to do. Okay. And one of the problems, something I also did not anticipate, because as the glue spread beneath the cover glass, everything was pushed to the side, and now they're also on the side here. That's a pity. That's a pity. Somebody also commented, but you know what? Doesn't matter. We're just going to repeat it. That's the whole point of a live stream. Not everything should be perfect, because sometimes you also learn things when things are not perfect. Okay. So what I'm going to do now is I'm going to use this one here. Okay. This is the one that I showed you at the very beginning. And there are so many of them in here right now, so that I think it should not be a problem to do that, because I'm quite sure that at least some of them will stay in the middle. Okay. So one of the things, for those, I mean, I did not tell you yet, but I did, wow, back in the day, I did study microbiology at the University of Vienna in Austria. So I've got a degree in microbiology, and the experiments that we did there, I would say that half of the experiments during my master's thesis did not work. Because we always had to optimize the methods. Sometimes there was a small mistake and the whole experiment didn't work. Yeah? So I'm going to put it on here again, and yeah, it's going to spread again. I'm kind of course, concerned now that whatever was in here is also going to be spread to the side. We can actually see that, but maybe we're lucky that some of the things will remain a little bit in the center. Okay, and let's try it again. I need to... Yeah. So if you're interested in going into research, then you gotta have a lot of patience, because see, in the center again, everything was pushed to the side, but at least here this one is still in. Very interesting to see over here is the following. Let me see if yeah, haha, isn't this beautiful? So this is also a nice example of what DIC is good for. Yes, it is pushed to the side, but it doesn't matter. It is still beneath the cover glass. And notice as it's moving, you see that the water and the glue are kind of mixing, and this changes the refractive index, and this gives it a little bit of some rainbow colors right around it. Yeah. You see, most of them are actually still pushed to the side. Yeah. So you see that the glue and the water, they are mixing, and yeah, the glue is slowly going to dry out, and look, here is another one of those things which is hatching. Yeah, some comments here. Yeah. Wow, there are lots of comments now. What is a permanent slide? Does it make it last? Oh, okay. I got some very important general questions here. So there is a channel called. Okay, I'm going all the way back in the comments now. Okay, that's actually the most common method. They're used to feed very small critters, most fish. Yeah, that's about the feeding. So hello here, Michael. Do you know whether the glue changes the refractive index of the water? Yes, of course. No, the glue will have a different refractive index, most likely. And for this reason, of course, the glue might not be suitable for some specimens. Why are you using glue? The reason why I'm using glue is for several reasons as a mounting medium. Because it is cheap, readily available, and also because it is water-based. Look what I have here. Because the whole issue about making permanent mounts is a science on its own. I've got here U-Pearl mounting medium, which is used by insect folks, entomologists, so those people who basically prepare insects, they use this very commonly. You got to buy it somewhere from a special shop. It's not so cheap, right? So that is a problem. And the thing is you're not able to put a moist specimen directly into U-Pearl because it's alcohol based. So there's a solvent in here and water and this, they don't mix very well. Other people have also used nitrocellulose as a mounting medium. Nitrocellulose is, you won't believe it, clear nail polish. And even that works, and even that has also been, I know someone who's been also doing microscopy research on amoeba, on the shells of amoeba, and he said at university they used clear nail polish as a mounting medium. Apparently because it had the correct refractive index and it dries very quickly. But there's a problem that it does not mix well with water. So if you add those to those critters, it's not. Yeah, it's going to be. There's going to be a wet water bubble in the middle, and then there's going to be the mounting medium around it. It doesn't mix, and then it can actually start to decompose and spoil. Yeah, and you don't want that. Yeah, so the mounting medium actually is also there to preserve the specimen. Yeah, so there are many of these mounting media around, but very few of them that are actually water based. And you can use also glycerin jelly. Some people use, but very difficult to use. You have to heat it up and so on. So short answer: why am I using the glue? Because for general purpose and for hobby purposes, it's the cheapest, easiest to use. Okay, I just had to say this because this is actually an important point. There's a channel called Macrofying that's getting lots of views and subscribers from photoshopped videos. Oh, okay, interesting. Yeah, permanent slide. Yeah, there's also quite a lot of videos going around saying the dust from the air landing on food is from dirty factories. Honestly, the dust from the air can make, I think, a whole different video. Here it is, a whole different video just of dust. You won't believe it's not just from dirty factories. You won't believe how interesting dust can actually be. Yeah, it can be really interesting because there are pollen in there. Of course, dirty factories as well, no question. Sahara here in Europe, sometimes every now and then, when there's a sandstorm in the desert in Africa, it's even carried over Europe. Yeah, so it's quite interesting what you're able to find in dust. Yeah, what is a permanent slide? Does it make it last longer? A permanent slide, yes. I have a whole slide box. A permanent slide is a slide that you can store for, if you make it well, for 100 years. I'm not kidding. Some permanent slides have stored for a very long time. You take the slide, you put it under the microscope. However, of course, there is no movement anymore because everything's been properly preserved. Yeah. So this is the thing. So does it make it last longer? Yes. And if properly done, significantly longer. Yeah, but again, of course, not for living things. And these ones here, I'm going to be honest with you, they will die in the next couple of minutes as the glue will start to dry up. And by next week, I would like to see how well they are actually preserved. Yeah, so that's a little bit the experiment that I would like to do. And next week, we're going to have another look at those. Yeah. Yes, that's the idea. Some of these permanent slides are nowadays over a century old. Yes, that's exactly what I just wanted to say. I've been reading it a little bit too late. Yeah. So it entraps cells or unicellular organisms. Well, whatever you put on here. Basically, instead of water, you're using the mounting medium. And preparing a permanent slide that lasts for a very long time is is almost a science. Okay, because what you have to do is you have to make sure that the mounting medium and the specimen are compatible with each other and that it's properly preserved. Maybe you can still move the glass to the side gently. The idea is maybe to move it a little bit so that I'm able to get more of them in here. I'm kind of worried. I could try to do that, but I think you should not do that because as soon as you've got something there, and if you move it, you're adding shear forces, which might actually destroy it a little bit. But why not simply just show you what happens? I need to get some tools here just to show you what happens when you're moving the cover glass now. Okay, I'm just going to get some pointed scissors, okay, to move the cover glass, and now you're able to see what happens. So I'm going to, I'm going to go down with the magnification here to flip it over here. I'm going to flip this out. And what you see here now is the edge of the cover glass, okay? So in the top corner, that's the edge of the cover glass, and you see that most of those, they stop moving already because the glue starts to harden out. Okay, and I'm going to simply touch it now and try to push the cover glass simply so that you see. Look, that's basically the tip of my scissors. Okay, so you see how big it is, the shadow at least of the tip of my scissors, and I'm going to now do the following. I'm going to move it a little bit, and now when I touch the whole thing. Yeah, then you can see that the whole thing does move a little bit, but yeah, it's still liquid. Yeah, but I don't think that this is a very appropriate way of actually getting them into the center. I think, what I could have tried is maybe waited until they're completely dry before I added the glue. But what I'm going to do is I'm just going to leave it like this, and I'm just going to, what I'm going to do, I'm just, after this live stream, I'm going to prepare a few more slides so that I have a few things to show you next week. Okay, simply to see how well they're able to be preserved. Okay, so yeah, I'm going to go again through some of those questions here. Maybe you can move the glass slide. Yep, and as you see now, there are the multicellular organisms. I quickly go through. Glue mounting media deserves its own chapter. Yes, indeed. It's also known as PVA mounting medium. Can also be commercially bought, but expensive. Yeah? So laboratory grade PVA. But then I found out that the glue contains pretty much the same thing here. Yeah. Do you have a recommendation for permanent slide set? No, well, let's put it this way, no specific recommendation. I just wanted to tell you that the quality of the permanent slides really goes apart, okay? So with quality, I mean sometimes the slides are kind of dirty, you know. You see that the people who made those slides, the companies, they did not really spend a lot of time actually cleaning it. So I see a lot of dust and dirt and other things inside the slide. Yeah, which is somewhat a pity, I think. Yeah, but then these are a little bit cheaper. Now there are some companies that actually make permanent slides, which you can buy individually for educational purposes and also for medical studies. So if you, for example, would like to have a cross section of the different tissues of the human body, different organs, you can buy all of this. Okay, because the med students, they need those, and those slides sometimes can be individual slides can be quite expensive. Okay, so do I have a recommendation? Not a specific one, I have to tell you. I've seen the quality goes apart quite a bit. You can shop a little bit on Amazon, but it doesn't matter. I highly recommend that you buy yourself anyway. If you don't have one, to simply buy one anyway, because there's simply lots of things to look at. Okay, maybe drop the specimen into glue drop, which is already on the slide. That is also something that I have done before. So the suggestion is the following. I just show you here, but the problem is that I cannot do it here so easily. Okay, so the suggestion is to add first the glue and then drop in the specimens. And this can be done if the specimen can be held with tweezers. Okay, then I usually take the specimen and drop it into the glue and add another drop on top and then the cover glass. However, the problem here is that I cannot pick them up individually because they're too small. Yeah. So this is a little bit a problem. Yeah. Is the carapace of these shrimp made of chitin, like other crustaceans should be, if they're crustaceans? So that's the carapace. That is the outermost, the exoskeleton. Okay. Nitrocellulose is gun cotton, which is rather explosive. That's actually correct, but I read some nitrocellulose. Nitrocellulose is indeed, I also heard that it is explosive, but I will research this. It's some kind of a, I'll research this, what it is. Maybe I switched the terms around. It is some form of a cellulose, which basically is in this nail polish. Nitrocellulose, but actually you're correct. This is actually an explosive thing. And but I know that old film also has been made of a similar substance, which was kind of flammable. Maybe I have to correct myself. Okay. Microscopy, does the glue also work for larger specimens like Daphnia, or small? Yeah. The thing is the following. The question is does this glue method also work with Daphnia? I've got Daphnia here. You know what? Let's try it. Okay. Generally, the issue is does the mounting medium infiltrate and is it able to go into the specimen to preserve it? That is the fundamental question. And normally what you have to do is before you mount it, you have to preserve the specimen. There are different ways of doing that, like for example, alcohol, which withdraws the water, it denatures the proteins, preserves it, and then you put it into mounting medium. However, the problem, especially with water microorganisms, is that the alcohol shrinks them up and shrivels them up. I think, however, that if you're using water fleas and other water crustaceans, because of their hard outer exoskeleton, the exoskeleton itself is not able to shrink so much. Okay, so what I'm going to do now is let's try to take up some of this here. I'm looking for a pipette. Okay, so these are now Daphnia. And look, the pipette, I've cut the tip because the Daphnia are yet larger. And now, up, let's take it with a little bit of luck. No, there's nothing on here. Ah, it's still in here. Okay, so you know what I'm going to do is I'm going to simply remove this water again. Okay, and here it is. Okay, there's a little dot here. Is Daphnia. I better not forget to show you the adult sea monkeys as well. And where do I have some tissue paper? Oh my gosh, I don't have tissue paper. Just a second. Here it is. And I'm just going to wipe everything here. And I simply discard the excess water. And, let's try to make a permanent slide of this here. I know some people will now write comments again and complaining that essentially I'm making a permanent slide of some of those microscopic water organisms because of animal rights reasons. Some people don't like that. You just have to read the comments in the YouTube channel. However, I will tell you the following, that they are going to die anyway because they're going to be eaten up either by fish. Yeah. So here it is. Okay, so this is, I need to focus this again. So that is Daphnia, and last week I made a video, a live stream of this, and also in my other YouTube channel I talked about it. And what you're able to see here in the back of this Daphnia, there are even some eggs. Okay, and what is over here next to it? So I don't know. I have to find it. It's kind of difficult to find. Is there something? What is this here? No, it's still the same one. Ah, look at this. Baby. A small one. Okay. Yeah. So the problem, I see that it does not make a lot of sense to make a permanent knot because it is way too thick. Okay. So this is not suitable. So what I'm going to probably do is I'm going to take away the large one, this one over here. I'm going to put it back, and I'm going to try to mount the small one that I have over here on the side. Where's the small one? Somewhere. This one over here. Okay, because this one actually is small enough so that it can, so that it makes sense. So it can be completely surrounded. Because otherwise, I have a millimeter thick layer of glue. It's going to take ages to dry. And it's also, not ideal. So what I'm going to do is the following. I'm going to take this away again. I'm going to remove the Daphnia, the big one. I'm going to put it back. Where is it? Here's the jar. And picking it up with tweezers is possible. You simply allow the water. To take it up, and I'm going to carefully move those into the center. So it might damage them a little bit. So let's check again. Let's close this here first. Let's check again if this is gonna work or not. Oh yeah, I always have to change over to the microscope view, and I have to find it. Here they are. Okay. And I'm going to mount them now, okay? So this means a drop of glue goes on top, and then the cover glass. So where is this? And I'm going to also keep them until next week because I would like to see if this is a feasible solution. It's way too wet. I just see that there is way too much water here. So let's remove some of this. I know this appears to be a little bit improvised, but if there's too much water, then this is also not ideal. But then again, if it completely dries out, it might start to shrink too much. On the other hand, it might start to shrink anyway when the glue dries. Okay, but let's give it a try. Yeah, now it's difficult to see where they are. And again, a little bit the same worry as before, that the same word that basically they are going to go all the way to the side, and I cannot see them anymore. Yeah, because of the refractive index of the liquid and of the Daphnia. I don't know. Honestly, I really don't know where they are. I see them. Yeah, it's being pushed to the side a little bit. It might work. Okay, let's have a look here. Microscope view again. Here it is. Okay. So here it is, and now I can go up with the magnification a little bit because there is a cover glass on top. Yeah, and up, it's here. I bumped into it, and then let's hope that it's going to work out properly. Okay, so this is okay, not a sea monkey, but a water flea, a fresh water flea, and we're gonna see how this works. So probably again a lot of questions here. Flash paper, maybe they are chitin. Negatives from film photography was made of nitrocellulose, exactly. Gonna build, that's why some of those films were also kind of flammable. Gonna build another slide ringing table project sometime soon. Okay, 3D printed one. Yeah, glad I made it for the stream. If you want to see the beginning of the stream, it's going to be, of course, available later. Maybe wait for the glue to dry a bit before using the cover glass. The critters will probably not move so easily in the edge. That could be. That's something to try out. Are those round black dots? Yes, these are eggs. Okay. So I'll quickly go down here if there are any more questions here. Hello from Northern Ireland. Hello. Maybe try putting the glue on a little away from them. That could also be possible, not directly on them, but maybe next to it. And then basically as you put the cover glass on it, the glue is pushed by the cover glass. That could be a possibility. Yeah. Here, pack them up with a drop of glue and a needle. That's also possible. I've also done this. Put some glue on the needle and then try to pick them up and transfer it. Yeah. So, okay. Is this under? Yeah, that's an Olympus microscope. That's correct. But so, have you done this method with tardigrades? No, I have not made. I've not made permanent mounts with tardigrades yet. Okay, so this I've not tried yet. So nitrocellulose in nail polish, nitrocellulose, the main film performing agent that varnishes, is used as a powdery fiber solid. Okay. Daphnia can't be moving. Well, the Daphnia is moving, but because I put it into glue, the movement is limited. Okay. So yeah, they're talking about nitrocellulose, which is the thing in the maybe the thing in the nail polish as a mounting medium. Okay, how they preserved? Do they change over time, discolor or anything? The thing is the following: if you are staining them, it is quite well possible that the specimens will change over time, especially specimens that are green like algae look much nicer when they're fresh. So I've got some permanent slides of algae, and the green, they're not quite as green anymore because pigments will lose the color over time if you have them, or some pigments will lose the color over time on the slide. So it depends a little bit on the chemical stability of the different pigments in the specimen. So I'd quickly continue to read the comments here. I would apply the glue on the side and the cover glass from the glue over the specimen. Yep, not tried that, but basically this prevents the specimen from being pushed to the side. I wish I knew of a mounting medium that was like sugar dextrose but more solid. I think must seem great. Yeah, there are so-called, there's also a mounting medium called fructose mounting medium, which is however a little bit acidic apparently, and therefore is also able to preserve some specimens quite well. How do the Artemia eggs look under a stereo microscope? Do the egg surface have any structure? They should have. I tried it. However, I have to tell you that the quality wasn't so good. But you know what? Where is my... we're going to try this now. Thank you for the comment. What I'm going to do now is, this is why I like those comments here, because sometimes I get ideas that I normally wouldn't have. So what I'm going to do now is I'm going to put some of those eggs and salt directly under the microscope with light coming from the top, because I'm going to use a flashlight. Okay, so let's try that. So over here, a small amount of those eggs in dry form. Again, I didn't try this, so I don't know how they look. A small amount. So there's salt here and hopefully some eggs. So what I'm going to do now is I don't know, there should be some eggs here as well. So what I'm going to do now is I'm going to put without water, I'm going to put a cover glass on top, simply that I don't spill the salt and the eggs. That's simply as a form of protection. So I'm going to just take one of those cover glasses here. They're unfortunately always a little bit dirty, so I give them I'm quickly going to dry wipe it. And I just drop it directly on here simply as a form of preventing the salt and the eggs. I hope I caught some eggs here as well. And under the microscope, switch over again. So this is not a stereo microscope, but the view should be very similar to a stereo microscope. Okay, so what you see here is this is now light coming from the bottom. Okay, so you see the salt crystals, obviously. Okay, and in the middle you see the round structures. But what I would like to do now is I would like to switch off the light, and let's have some light from the top. Okay, and then I might have to refocus a little bit. It's not very, maybe I can. So, actually it's quite nice. Look. This is how they look like. Some of them actually look like they're indented, like. Yeah, so you see the white salt crystals, and you see the brown eggs, which many of them are not round, but kind of, I don't know, like bent inwards, like a ball, which doesn't have enough air. See, that's one of the nice things about the live streams and comments, because I didn't think of doing that. Oh, the flashlight went off because the battery is already bad. It's a rechargeable flashlight, and apparently I didn't charge it. Okay. But I think you got the idea. Yeah, that's unfortunate. This flashlight here is a rechargeable flashlight. Yeah, so there's a charger over here, but it goes out. It has 2 intensity. Yeah. Yep. That's a pity. Okay, but I think you get the idea. Okay, so it is possible to also look at the things using light coming from the top. Wow, lots of comments again. So I'm going to, how do the Artemia eggs look under the stereo microscope? So this was not a stereo microscope, but it was kind of similar, okay? So mycelium looks great in a petri dish, but would be better as a permanent slide. And mycelium is from a fungus, okay? Can you show the glue bottle again for the camera? So so yeah, I can show you the glue bottle. Where is it? I bought this over Amazon. It was pretty cheap, really. It's called Elmer's glue. And it says washable and child-friendly, but you have to be careful because they have several types of glues, and most of them are white. Okay, but this one is the clear one. Yeah, so it's Elmer's glue, and yeah. So you see what 83% natural ingredients, school glue. Yeah, and basically the reason why I recommend it is because mounting media that are normally used for microscopy, like if you really want to make slides that last forever, those mounting media are kind of expensive, and you have to make sure that the specimen is completely dry, and most mounting media that you can buy are based on a resin-based mounting media that contain poisonous solvents, not even alcohol, but xylene and so on, and which you don't want to have in your household. And for this reason, they're not recommended. Euporol is one of those rare mounting media where you can transfer the specimen directly from alcohol. But then again, if the specimen is delicate and moist, especially for water organisms, then the alcohol is a problem again. So that's the reason why I ended up with glue here. And if you put nail polish directly on a moist specimen, then it doesn't mix at all, and then you have clouding happening. So basically it doesn't mix at all, and then it looks pretty bad again. Yeah. So for this reason, if you just want to play around a little bit and experiment, especially if you maybe are also in an educational setting or so, and you want to have it safe, and the demands on the permanent slide are not that high, if they basically they don't have to last 100 years, yeah, then I would say just play around with this first, and if you're happy with this, then it's fine. If not, then you need to buy yourself anyway specialized mounting media. Yeah. Is Elmer's glue, but any PVA glue should be okay. Yes, yeah, because this is contains also PVA. Yeah. Yeah. Almost. Yeah. Quickly scan through the comments again. What's your opinion about Delphi X Observer Trinocular Microscope? The Delphi X from Euromex, right? Okay. The Delphi X, I think, is this not the microscope from Euromex? I think it's a pretty good one. I think the Delphi X, if I remember, even has the options to come with DIC optics, because I actually several years ago I actually had a look into this. Yeah. I wonder if shrimp would incorporate the antioxidant found in some algae and what makes salmon red. I would make it okay. I don't know. That's an interesting one. Yeah. I don't know about that. They're dried out. When they're placed in water, they suck up the water and inflate. Yes. Okay. Okay. Just for the patron, just to become to buy flashlight batteries. I wonder if Limaline would also work. I don't know. Yeah, could be, but I know that some of the mounting media already contain the solvent xylene. So, for example, one of the mounting media that I used was UKIT, which is a very fast drying one, but it also contained some solvents that are not so healthy. Yeah. If you use white glue, would it still work because it turns clear? I don't know. I've tried that. I've not tried that. Okay, so I know that white glue turns clear after some time, so that would be something to try. However, if you use white glue, then you don't really know. Okay, you cannot see where the specimen is at the beginning until it's clear, and this might be a little bit of a disadvantage. But I would say that's something that one certainly could try out. Yeah. Okay. Yeah. Aren't there also rotifers used for fish food? Why not? I know fish definitely eat rotifers, but I don't know if they actually are also sold as fish food. Yeah. So what I'm gonna do now is, wow, I'm already over an hour, but I promised that I also wanted to show you, at least those of you who were not here last time, some of the adult sea monkeys, and I think that's gonna be one of the last things that I'm gonna do now, because I've been already talking for over an hour. But I recommend that you try this out yourself anyway. So again, I'm going to use now the large one over here. Yeah, it's normally you should not, for contamination purposes, you should not mix different water samples, but honestly, who cares? So, and there are not quite as many of them in here, but maybe I'm lucky. And 3 of them. I remove any excess water, and I'm not going to add a cover glass because I do not want to squash them, and then later on so that I can return them. And yeah, let's have a look here. Again, those of you who joined my live stream last week have already seen them before, but these are now the sea monkeys, the Artemia brine shrimp in adult form. So where are they? And they're huge. Okay, so I won't even be able to fit them. Yeah. So that's one. That's the other one, and maybe there's too much going on here. That's the third one over here. It's a little bit out of focus. Yeah. Yeah, but you see they're gigantic. Yeah. Yeah. So these are the ones here, and they are, I would say they're all a centimeter long, almost 8 centimeter, about this size. Yeah. And I'm using the lowest magnification objective, the 4 times right now. It would of course be, you know what, I'm gonna try it anyway for a few seconds. Where is my flashlight? The one without the battery? Where did I put it? I've got so many things standing around here that I don't even know where I put my, here it is. So maybe if just for a couple of seconds we're able to see this. Yeah. Yeah. Not very bright, huh? No, I think this does not look very good. Okay. Now I think we're just gonna use the light coming from the bottom. Yeah. And I just wanted to show it to you to show you that they yeah, not only are different in size but also completely different in yeah, in appearance. By the way, if you look at the water crustacean, the shape of the larvae, they look very similar, right? Even the shape of the larvae of Daphnia and other water crustaceans, they look quite similar, but then the adults sometimes they look quite different. Yeah? So I go through here again. Did you put them under the microscope? I don't now know what you referred to. Yeah. Yes. Oh, the one from the Euromex. Yes, it's from Euromex. And do you mean Delphi X Observer trinocular microscope come with DIC? You can. I think the Delphi X Observer, if you download the pamphlet, you can actually upgrade it to DIC. I think so, because that's actually yeah, one of the microscopes that I had a look at. Yeah, but I mean, it's not cheap. Huh? And I grew dweller algae to feed to the rotifers. Yep. Got a fish tank. Do you stream? I love to waste 40 minutes a day looking at fish. As a matter of fact, what I tried before I started to do those regular live streams where I'm talking, I actually tried the following. I actually put a microscope slide with some water microorganisms under the microscope, and I simply connected the camera and simply streamed whatever happened to be on the microscope slide. But there is a problem with that. The slide dries up very quickly, and yeah, it's not very suitable for continuous live streaming. And I do not have a fish tank. I used to have one about 20 years ago, but not anymore. Yeah. How long does it take for the larvae to mature to adults? I don't know. And I think there are probably several stages, because I think they molt, they kind of shed off their exoskeleton. So I think that, yeah. And what I also read is it depends quite a bit on the temperature of the water and the environment and the amount of food that they get. But honestly, I don't know. And that's something that I would like to try out anyway. I would like to see if I'm able to grow some of those larvae all the way to adults as well. I have not grown those to adults. I got them already finished from a shop. Yeah, there was something moving over here as well. Where is this? Let me quickly check. Just yeah, it's you see it's here. I see. Here's a small one. Why is there a small one there? Did they reproduce, or is this some kind of a contaminant from the pipette? Now that's interesting. Okay, because there were actually only adults. I bought we bought one with only adults, and so kind of wonder why all of a sudden there's a small one in here. So it could have been maybe something from the pipette. Yeah. No, I did not. I ran out of time. Research project. Okay. What are those appendages in the adults? There's so many. Yeah. Lots of appendages here. Neat to see them change. They can get really big, and many have impressive mouth parts. Yeah. And two eyes as well. Yeah. Unfortunately, no. When I move soon, I will set up a little lab and tank and outside ponds. Yep. Yeah. Artemia, I think. Oh, yeah. Somebody, I think, who joined later on. These are Artemia brine shrimp, and they're also sold for children with the name sea monkeys. Yeah. Because Artemia brine shrimp does not sound very exciting. Oh my gosh. Are these? Look, these are eggs. They are reproducing. Look at this. Okay, here we go. Okay, folks. The lucky coincidence. Yeah, this one over here. Yeah, seems to be producing eggs here. Let's make it a little bit brighter. Yeah. So this also explains a little bit why I was able to see this one larva, because I was kind of wondering whether it was a contaminant. Evidently not. Evidently it is. They are reproducing in this little jar here. That's kind of nice. Huh. Can you seal the cover glass with Vaseline or something similar to prevent the slide drying out quickly? Yes, Vaseline, however, is normally used to not necessarily for sealing it, but for gluing on the edges, okay? For gluing the cover glass to the slide so that the microorganisms have a limited vertical movement. It's not used so much to prevent them from drying out. If you want to prevent slides from drying out, I recommend that you make yourself something like this. There is a little, it's a plastic container. There's a little rack in here and there is water on the bottom and you put the slide here on the top. Okay, and when you do that and you put the cover, the lid on top, then the slides will not dry out and you can keep them for a fairly long time. I think that's a better way than working with Vaseline. Yeah, or you keep on adding a little bit of water. Okay. So, can you seal the cover glass? Yeah, so it will reproduce quickly as soon as males and females are together. Okay, you have young ones. I think the females carry the eggs until they hatch, almost live birth. Yep. You see over here. Here they are. Okay. I'll watch if you do. Okay, have a great weekend. Okay. Yeah. One can make a humid chamber for slides. Yes, that's what I essentially just wanted to talk about here. That's a humid chamber. I use plastic container. What's your most expensive microscope? Well, I've got several microscopes and I would say that I'm not going to talk about the price now, but I would say that any microscope in the range of, let's say, a few $100s or euros essentially will produce results like this here. So you do not need a lot, and the magnification is not important. Not so important, because they magnify pretty much all the same anyway. Yeah, but that is of course a nice example here. Okay. Yeah. So yeah, here's another egg. Okay, what time is it now? 1 hour and 21 minutes. I've already been talking here. So what I'm going to do now is I'm just going to slowly call it quits. For those of you who joined in late and who were kind of wondering what the whole thing was about, just a short summary. I basically got some brine shrimp eggs here. These are brine shrimp eggs, and it's a powder which contains salt and eggs, and when you mix it with water, with the correct amount of water, then you're able to be able to breed them. They are sold as fish food, but they can also be bought in kits for children, so that you can make yourself a home aquarium. However, I think that those brine shrimp, as fun as they are, especially after they hatch, they are very, very small. You do need a magnifying glass. I have over here a magnifying glass to look at. So to look at them, and then you can actually see them swim around. But I think that if you really want to see them well, then I think they're a nice specimen also to put under the microscope. Especially some people want to keep themselves motivated. They don't know what to look at. They want to do a little bit of nature observation. Some children, maybe with parents and children, and in schools maybe. And I think that this is a nice example which can be combined quite nicely with microscopy. Yeah, and. So basically it's a fairly simple thing to do, and if you're lucky, and then you can also grow them until they're a little bit larger. I also showed you that it's possible. Oh, I did not show this to you yet. Okay, you know what? Let's do this anyway. Let's feed them. Just very quickly, so that you see how this looks under the microscope. Okay, so I'm going to quickly feed them. All I have to do is I'm going to take just a slide. Okay. And I do not want to add too much of this. I'm going to quickly switch over, okay? And I'm going, where's this? Okay, I'm just going to add a drop here, because I found out that this is so concentrated that essentially, it's not a good idea to have too much of this. So I'm going to take a small, tip here. I don't know, it's almost nothing, and I'm just going to add it to those here, and then you can actually see how the food spreads. And this also allows you to see a little bit how the water is moving. So I'm going to touch it now. Where is this? See all of these green little dots that you see, these are algae of the food, okay? And yeah, this is basically the official food which is made specifically for them. You can go up maybe a little bit with the magnification. You can actually see that it does look a little bit green, and they're going to eat that. And I read in the instruction that the particles are small enough also for the larvae to eat them, because size matters in this case. That if they're, and yeast cells are large, or maybe probably too large, okay? So maybe that could be a disadvantage for the young ones, that maybe the yeast cells are too large, but those algae or algae fragments or whatever this is actually is suitable as a food for them. Yeah. So it's you see it's quite dense here. Yeah. Yeah. Just wanted to also show it to you because I almost forgot about this. Yeah, there was the small one over here again. Yeah. So yeah, so I'm slowly going to finish now. Okay, I could just quickly go through the remaining comments here. Okay, sharing all of this has been a delightful way. Thank you very much. Okay. What are these objectives? You talk about the microscope objectives. Well, they are part of the microscope that I got. Yeah. So how do you look at plants under the microscope? You know what? This here are plant parts, 25 years old, preserved in alcohol. You have to cut them very thin. That might be the topic of another video, is how do you microtome plant parts? I could do that, okay? Can you do, would a live stream about algae be ideal, or would that be really boring? Depends on the algae. I mean, I got a few containers over there with algae. I can show them to you. But it's a little bit of a question of luck what you're able to find. Yeah, lots of diatoms. Yeah, and some of them they're pretty repetitive. But it's something I can, in any case, show you in the future. Okay, the wet algae are mainly for the young ones. The adults can be fed with dry algae. Okay. Would you be able to use this glue also for botanical preparations instead of Uperol? Uperol is ideal for insects because it clears up the exoskeleton. It makes the insects more transparent. I have made also botanical preparations with Uperol before. However, it takes around 6 weeks for the Uperol to dry. 6 weeks around. Okay, so the drying time is very long, which has the advantage that it gives it enough time that all parts of the specimens are actually covered. Yeah. What will you show next time? I don't know. Well, I'm going to show you the slides, okay? The slides that I made today, so that we can actually see how well how well it actually worked in a dry form. But I don't know yet. So what I've done is I've added a little poll, a questionnaire, what you're mostly interested in. And so far, most people said they're interested in specimen preparation. So maybe I'm going to put a focus on that. Okay. If you are basically if you're new to this channel here, I do have a second channel where I'm uploading shorter videos on a more regular basis about different things that I find under the microscope. So you might also check the main channel. This is not the main channel. This is just the live streaming channel that I have, and where I talk about technical aspects. But I also have a main channel where I'm putting more specimens under the microscope. Yeah. Okay. Feed adults hobby microcellpulver. Oh, okay. Yeah. Please tell me how to preserve the plant parts for a longer time. Generally, if you want to preserve any specimen for a longer time, make sure that it is mostly free of water. Okay, but if you make it free of water, then there's always the problem that it starts to dry up. Okay, so what you have to do is you have to then immerse the plant section that you want to preserve in a water-free environment like alcohol, and then transfer it into a mounting medium that contains alcohol. Or, and that's the other thing, is you use this glue and you allow all of the water to evaporate. Then it's also very dry. Okay, so that is a little bit the thing. For long-term preservation, you need the specimen must be free of water. And the reason is because as soon as water is present, then a bacteria can grow and it starts to decompose, and then the slide is not stable over a long time. Okay, people, I'm going to do the following. I'm going to call it quits for today. Hope that you liked it. I hope that these videos also motivate you to try this similar things and to experiment yourself a little bit. If you have any suggestions on what I could put under the microscope, then please do mention it. Many of you have already done that. There's also this little poll that I have, a little questionnaire of what you're mostly interested in. I would say all the best to you. Hopefully see you again next week. Happy microbe hunting as always, and bye-bye.